So in any circulatory system, you have to have a fluid medium that can carry your gases. You have to have a pump that creates pressure to move those gases through the body, and you have to have some sort of vessel to contain that fluid and direct where it goes. Now, in our discussions of the circulatory system over the last couple of weeks, we've talked about the medium, blood, and we've talked about the pump, the heart, and now we're going to talk about the vessels. And there are three main types of vessels in your body. There are arteries, which carry blood away from your heart. You have veins, which carry blood back to your heart. And you have capillaries. And capillaries are probably the most important of these vessels because that's where exchange happens, right? So capillaries are made of very thin-walled, like simple squamous epithelial walls that allow diffusion of gases and other substances back and forth between the fluid around your tissues, your interstitial fluid, and the blood plasma. And your circulatory system really has two circuits. It has a pulmonary circuit, which goes to your lungs to pick up oxygen and then back to the heart. And then a systemic circuit, which goes from the left side of the heart out to all of the tissues of your body and then back to your heart. So blood comes from the systemic circuit to the right side of your heart, then from the right side of your heart through the pulmonary circuit to your lungs and back, and then from the left side of your heart into the systemic circuit and back. We are going to talk about a lot of hollow organs and tubes this semester. We've already talked about the heart, for instance. And in most of these hollow organs or tubes, you're going to have at least 3 layers of tissue that you find. Generally, the inner one is going to be some sort of epithelial or endothelial tissue. The middle is going to be muscle, and the outer layer is going to be some sort of connective tissue that wraps it all together. In the case of blood vessels, arteries and veins, at least, are going to have a tunica intima, which is this inner layer that is made of endothelial cells. There's a tunica media in the middle, which is made primarily of smooth muscle, and that's important because that allows your vessels to get smaller or larger, really narrower or wider, and that helps regulate blood flow. And on the outside of that blood vessel, you also have a tunica externa, which is sometimes called the tunica adventitia. And this is just a sheath of connective tissue that packages it all together and anchors it to the tissues that are surrounding it. So the inside is the tunica intima, or sometimes it's called the tunica interna. The middle is the tunica media, which just means middle, right? And then the outside is the tunica externa, or tunica adventitia. And while both arteries and veins have the same 3 layers, they look a little bit different. And if you're looking at them under a microscope, you can see. So arteries have a much thicker layer of muscle than veins. And they also have much more elastic tissue in there, many more elastic fibers. And this allows them to expand and then regain their shape when there's a change in blood pressure. And this is important especially for the largest arteries that are close to your heart. So think about your aorta. Your aorta has to deal with a huge difference in pressure between systole, right, when the ventricle pumps all the blood out, and diastole, when it's relaxation. So it has to be able to deal with that pressure and then snap back to its original shape, hence the muscle and the elastic tissue. But because of that muscle and elastic tissue, it also tends to keep its shape. Arteries tend to be round. Veins are thinner in the tunica media and are often sort of collapsed. They don't necessarily have that round shape when you look at them under a microscope. And because of this extra elastic tissue, those largest arteries, like your aorta and your pulmonary artery, those ones that kind of leave your heart and shortly thereafter, are called elastic arteries. And then the next largest arteries, which kind of go to each organ, specifically to each muscle, are called muscular arteries. These are medium-sized, and they have a little bit less elastic tissue, but still a fair amount. And those muscular arteries will branch and branch and branch into smaller and smaller arteries until you get to arterioles, which are very small. They're the smallest arteries, and they lead to a capillary bed. These are not good for gas exchange like capillaries, but they're so small that the tunica media, while present, is like 1 or 2 muscle cells thick. It's very small. So from your heart to your elastic arteries to your muscular arteries to these little arterioles, now we get to the capillaries. And capillaries are a single cell layer thick. These are very thin walls, and those cells are the thinnest possible epithelial cells. They're squamous epithelial cells. And this simple squamous layer is great for ease of diffusion, allowing water and things dissolved in it like sodium and glucose and amino acids and oxygen and CO2 and those things to diffuse and be exchanged across that membrane between the tissues and the circulatory system. And this is kind of the whole point of the circulatory system, right, is to allow for this kind of exchange. And now we enter the veins, the back end of the system, where we've already had our exchange. We have deoxygenated blood, if we're talking about the systemic circuit at least. It would be oxygenated in the pulmonary circuit. But we start with the very smallest venules, right? So arteries start big from the heart and get smaller and smaller and smaller as they branch out. Venules start small and then join one another, like little streams joining into creeks and rivers and then eventually to the ocean. So we start with this very thin venule collecting blood from the capillaries after gas exchange. These tiny venules come together and join into larger medium-sized veins, which are going to drain from different organs, and then these medium-sized veins eventually are going to drain into large veins. And these large veins are huge, and they are going to include the superior and inferior vena cava, which dump into the right atrium of the heart, and also the very large veins that kind of feed the superior and inferior vena cava on their way toward your heart. And this is a nice summary figure from your book showing you the pathway of blood in the systemic circuit, away from the heart through elastic, muscular, and then arterial arteries, and then into the capillaries, and then back through venules, medium-sized veins, large veins, and back to the heart. So let's take a deeper dive on capillaries. There are 2 main kinds of capillaries. There's a third that we'll talk about in a moment, but the first is continuous capillaries. And continuous capillaries do not have any pores in them, as we'll see. The endothelium here, these endothelial cells, all connect and they make a complete lining. And these are found throughout your body, and this allows this complete lining small things like water and tiny solutes and some lipid-soluble materials that can move across membranes to diffuse, but it prevents you from losing things like plasma proteins, larger stuff that can't move through there. And you also have some selective vesicular transport, which means active transport through the cell through vesicles, which is expensive, but it allows you a lot of control over what moves across these capillaries. So these are continuous capillaries. Conversely, as opposed to continuous capillaries, fenestrated capillaries have little tiny holes in them, or fenestras, which means windows. And these little holes allow more stuff to move across more quickly. So water can move really quickly across these. So can some larger solutes, although it still is going to prevent very big things like most proteins, blood cells, things like that from moving across. But again, these things allow for quicker diffusion and exchange, but they're less picky about what goes across. And these are found in a few different places in your body, choroid plexus of your brain, where you create your cerebrospinal fluid, but your kidney filtration sites are among the most important, and we'll talk about those when we get to the kidneys later this semester. So continuous capillaries are the most difficult for things, especially large things, to move across, but they're allow you a lot more control over what diffuses in and out. Fenestrated capillaries allow things to move more quickly and allow you some control. Big stuff still can't really move through easily. But you have to have some capillaries that allow you to move things like proteins and occasionally cells in and out of the bloodstream. Right? Your blood proteins are primarily made in the liver. And so you have to have a way to put them in your blood. So you have sinusoids. And sinusoids are capillaries with big gaps in between cells. where there essentially is no membrane, right? You have these large gaps between cells, and that allows you to put proteins in or take big things out. You don't find these in as many places, but you will find them, for instance, in your liver, also in your bone marrow, where your blood cells are created, as we talked about a couple of chapters ago. You have to be able to put your red and white blood cells into your blood, so you need sinusoids to do that. You'll also find them in your spleen, which is where red blood cells get taken out of commission, remember, at the end of their lives and eaten up by macrophages. While arteries or veins may run as like a single artery, right? You have your radial artery that runs down your arm and your carotid artery that runs up your neck on each side, right? Capillaries run in these beds, right? Giant networks of interconnected vessels, and a bed will run between an arteriole and a venule. Now, because they're so small and there are so many of them, this gives you a lot of surface area, right? And surface area is really important for exchange. That will be a big theme in this class too. So if you remember from last semester in 151, you would have discussed how, one large cube of a particular volume has a very small amount of surface area compared to its volume. But if you split that cube into a bunch of tiny cubes of the added all together at the same volume, you get much more surface area, right? If we had 100 people all trying to crowd around one large pizza, it would take a lot longer to eat that pizza than if you could cut it up into slices and hand them to each of the 100 people. Then, boom, 20 seconds and all of that pizza is gone. Same idea with these capillary beds. Lots of tiny vessels, lots of surface area for exchange of gases and nutrients and wastes. And these capillary beds are often supplied by more than one artery. Multiple ones are called collaterals. And very often those collaterals will have connections between them, and those fusions are called anastomoses, or anastomosis singular. And this is important because if you get a blockage in one artery, it allows blood to zip over to the other artery and still make it to that capillary bed, so you're still able to get blood flow there so the tissue doesn't die. And you can also avoid sending most of your blood to that particular capillary bed if you'd like, through an arteriovenous anastomosis, which is, again, one of those connections, fusions, that just connects a blood vessel from the arteries to the veins, bypasses the capillaries. Or a thoroughfare channel is an example of this that goes directly through a capillary bed, but does not fill the capillaries. And you have regulators that are called precapillary sphincters that are little tiny smooth muscles that open up when you want the blood to fill the capillary bed and close up when you don't want that blood to fill the capillary bed. And this is kind of what that looks like, right? Here's your thoroughfare channel, here's an arteriovenous anastomosis, and this is, your capillary bed. There would be a lot more of them. This is a very small one. But again, you don't have enough blood to fill up all of your capillary beds at once, right? Depends on what organs you're using, which capillary beds you want to fill up, right? If you are eating food and digesting it and absorbing it, you want a lot of that blood to go into the capillaries around your intestines. If you are running a race, you want a lot of that blood to go to your legs, to your quadricep muscles and your calves. I remember when I was in high school, I was in the Spanish club, and we did this trip to Spain that was wonderful, like a once-in-a-lifetime thing. And we had one day where we got to, instead of going to museums, go to the beach. And the guys all stayed in one room and the girls stayed in another. And my friend Rob, before we went to the beach, told the guys, he's like, we need to do some push-ups so we look strong. And I said, I'm a little behind on that. You got to do those for months, right, to look strong. And he said, no, you do it and you look big. And he was right, because when you do push-ups, you're using those muscles and your body fills those capillary beds with blood. And so they look bigger for a short amount of time, right? So again, we have a lot of regulation over what blood enters these capillary beds. You can see these tiny smooth muscles here, which we call precapillary sphincters, regulating that flow. And at the end, this deoxygenated blood flows over into the veins, again, at least in the systemic circuit, and the veins will carry that blood away back toward the heart. And in the next video, we're going to talk all about veins.